person. But as many of you know, Frank is one of the, I would say, even founders of the RAS MAP kinase pathway and even RASopathy research. He's currently the David Wood Chair of Tumor Biology and a professor at UCSF at Helen Diller Cancer Center. And Frank has kind of a different career than most academics because he actually did most of his career in industry. He was for many years the CSO of Onyx Pharmaceuticals, so he has a broad experience on drug development where they develop sorafenib. They also develop certain oncolytic viruses as well as palbociclib that later went into Pfizer, I think. On the research is also credited with the discovery of several important things, most importantly, perhaps gap activity. He discovered gaps in general, which explain a lot of the mechanisms of RAS mutations that they evade gap and therefore become active. He also went on and cloned some of these gaps like RAS-A1. He also collaborated in understanding the function of NF1 as a gap of RAS. And as you probably know, he's also had a huge impact on the discoveries related to RASopathies. So for instance, his group discovered the SHOC2 RAS PP1 complex. He's also done identification and characterization of CFC variants with Kate Rowan, LZTR1 more recently. So he has, I think, a very vast knowledge both on the basic signaling of the RAS MAP kinase pathway, but also how to translate some of these molecules. So with no further ado, Frank?
Thanks a lot, Pao. Can you hear me okay?
Yes.
Okay, great. Thank you. Yeah. Well, as Pao said, unfortunately, I came down with COVID a few days ago and I'm still viremic, so best not be there. But I really regret not coming to this meeting because I've attended many times in the past and have always learned a great deal from the presentations and from the interactions. And I could hear just from the buzz of people talking during the coffee break there that there's a lot of really interesting topics being discussed. And today, I'm going to try and summarize what we know and what we don't know about the sort of fundamental pathway at the heart of the RASopathies, the MAP kinase pathway. And I'll try and make the point that although this pathway has been around for a very long time, we still don't really understand how it works in molecular detail, and there's still many questions to be answered. And generally speaking, a better understanding of this pathway will lead to better therapeutics for RASopathies and for cancer. And that I'll attempt to share my screen. Okay, so these are my disclosures. And please, Power, if you can't hear me or something goes wrong, just text me. I have my phone ready for you. Okay. So this is the textbook version of the RAS MAP kinase pathway, which many of you will have memorized in high school probably. But again, I'll make the point that although the general flow of information down this pathway is undoubtedly correct, we don't know a lot about the details of how this whole pathway works. But just by way of orientation, you all know that RTKs activate complex of proteins involving activation of SOS to push RAS from the inactive state into the active GTP-bound state, where RAS can engage the RAF kinases, which activates MEK and ERK. So that's the sort of the fundamental flow of information. And you can see this flow, I'll just get my laser pointer going here. Okay, just by looking at sort of biochemical analysis of the pathway. So if we add EGF to cells within 1 minute, you see accumulation of RAS in the GTP-bound state, and by 5 minutes, it's pretty much gone again. And you can see activation of phospho-ERK a little bit later as information flows down the pathway. You can also see this pathway in this nice representation from John Albeck and colleagues, in which he's using a phospho-ERK sensor to measure pulses down this pathway. And what this shows very nicely is that in the presence of EGF, you see pulses of a signal going down the pathway. And the more EGF you add, the more pulses you get. And the point of this really is that this pulsatile signaling is what happens in normal cells. And each of these pulses is very, very symmetrical and very similar in size and duration to show that the whole pathway is extremely tightly regulated. in normal cells. Another one thing I want to point out is that in normal cells, the only effector of RAS proteins is the RAF family of kinases. As far as we know, RAS proteins don't do anything else in normal cells. And that might be a bit of a surprise for people who work on cancer. And as I'll mention in just a moment, in cancer, the situation is very different. But in normal cells, this is the function of RAS, to activate the MAP kinase pathway, and it's the only effector that does that in cells as far as we know. Now, this representation here makes it look as if each of these kinases sort of passes on a phosphate, maybe like a sort of baton or relay. But this is actually not the case at all. This is a very complicated high molecular weight structure. Many of these proteins are all bound together in one big structure, as I'll show you, RAF and MEK and possibly ERK, all associated together in a very complicated way that is regulated by other chaperone proteins and scaffold proteins to fine-tune this pathway accurately. So this is much more complicated than represented here, and we still don't understand precisely how RAS activates RAF, for example. The output of the pathway, if you're familiar with this pathway in cancer, you tend to think of it as a pathway involved in proliferation because the pathway turns on transcription of the cyclin D1, which pushes cells through the cell cycle, as you all know. But again, from studying RASopathies, we know that activation of this pathway, even mild activation in RASopathies, can lead to alterations in differentiation and development and even cognition and behavior. So the biochemical outputs of this pathway vary enormously depending on the cellular context and many other factors we don't really understand at all, at least I don't. Now, as I said, in cancer cells, things look different. And this is the kind of pathways you get if you just Google what the RAS proteins do. You'll see RAS proteins activate, obviously, MAP kinase, but also PI3 kinase, RALGDS, phospholipase C, other effectors. And there are various versions of this from the literature. If you ask ChatGPT, you get a slightly more accurate version. That is, RAS activates the MAP kinase pathway primarily, but can also activate PI3 kinase and RALGDS. So these additional pathways are only seen in cancer cells and not in normal cells. And I'll explain why that is. So at least this is my version of the way things work. So in normal cells, RAS proteins activate MAP kinase. The closely related proteins, RAS and RAS2, activate PI3 kinase. MRAS activates SHOC2 and SCRIB and maybe other effectors, and RAL proteins has its own set of pathways. So in normal cells, each of these proteins stays in its own swim lane, essentially, and activates its sort of private effector pathway. But if you have a mutation in the pathway that activates RAS and makes a mutant form of RAS proteins, then RAS can cross over and activate other effectors such as PI3 kinase, SHOC2, RALGDS, and so on. So this is the gain of function you see when you have a mutant protein overexpressed, stuck in the GTP bound state, picking up functions from its very close cousins where it's not supposed to interact. And just FYI, RAS proteins, wild-type RAS proteins, have a low affinity for PI3 kinase, about 20 micromolar binding. Our RAS proteins bind much more tightly, about 3 micromolar, and therefore engage it naturally. But mutant RAS proteins like G12D and V have a higher affinity for PI3 kinase than wild-type RAS, and they're overexpressed, and they're mutated. So they can cross over and activate PI3 kinase as part of their cancer biology. And similar arguments apply for other RAS neoeffectors. And this kind of crossing over of swim lanes can go both ways, essentially, because in RASopathies, mutant proteins that are supposed to be doing their private functions can now cross over and activate MAP kinase pathway. And that's how we think that RAS family members take on the activation of MAP kinase pathways when they really shouldn't be doing that in normal life. Get this out of the way. Now, the reason that this promiscuity is possible is because these proteins in these families are very, very similar. And particularly the effector binding region, which is this blue bar here, this switch 1 region of the RAS protein. The sequence of this effector binding region shown here, and it's identical between different members of the RAS superfamily. And yet only H, K, and N RAS directly activate RAF kinase. So we can ask, well, how about the RASopathy proteins, MRAS, RAS, RAS2, and RIP1, which have identical effector binding sequences? They don't activate RAF kinase directly. So how do they activate the pathway? And that's what I'll talk about today. So we have a better understanding of how RAS proteins interact with RAF kinases, and I'll get back to this more later in the talk. But the sort of bottom line for now is that RAS proteins bind at high affinity to the RAS binding region of RAF kinase. This interface here, again, is identical between all members of the RAS superfamily, and this is the high affinity interaction. When Samanchu and colleagues at Frederick solved a bigger piece of RAF, which includes the cysteine-rich domain here in green, now we see a bigger footprint of RAF on RAS. Now, these interactions, which are essential for activation of the protein, not just binding, but activation, these are unique to the canonical RAS proteins H, N, and K. So all RAS proteins can bind, but only canonical RAS proteins can actually activate because of this additional contact point, which is critical to the activation process, as I'll discuss later. Okay, so these are all the genes which you're all very familiar with, which are mutated in Noonan syndrome and other RASopathies. So green are gain of function, and red are loss of function. And there may be additional genes which I haven't included here, so I apologize, but this is basically a pretty good consensus. As again, you all know, when this pathway is somewhat activated, you see changes in development and differentiation for pathways which aren't well understood. But they are well understood in a kind of related disease, not technically speaking, maybe a RASopathy, but related disease, achondroplasia, which is caused by mutations in the FGFR3 gene to activate the kinase activity. FGFR3 is one of Anne Gorali's selfish genes that self-amplifies in the testes of elderly males and is responsible for this, predominantly responsible for achondroplasia. So this condition is caused by premature differentiation of chondrocytes in the growth plate of the long bone, as shown here, and the signaling of this is quite well understood. Now, when we started BridgeBio many years ago, we were able to license a FGFR3 inhibitor from Novartis. Infigratinib had been developed as a drug for treating cholangiocarcinoma, where it's actually now approved for treating cholangiocarcinoma, a tumor driven by gene fusions in FGFR receptor type 2, but the drug affects FGFR3. We performed a clinical trial to see if this could reverse the effects of FGFR3, and summarized in this Nature New England Journal of Medicine paper, the results are shown here. Typically, individuals with achondroplasia have a reduced growth velocity of about 4 centimeters per year instead of 5 to 7. But in the presence of this inhibitor, by toning down FGFR signaling, the growth velocity accelerates by an additional 2.5 centimeters a year to put it into the normal range. And the dose escalation in clinical trials, the FDA actually advises to stop at this point because we now have reached normal growth velocity. So a dose of this drug, which is actually 500-fold less than the approved dose for cholangiocarcinoma, does actually reverse the phenotypes of achondroplasia. So if this all pans out and the drug is approved, parents will have the option of avoiding most, if not all, of the effects of hyperactivation of FGFR3 in this disease. Okay, it's not exactly a MAP kinase, only disease. It's a bit more complicated, but it does make the point that drugs that being developed for tumors can have effects in RASopathies at much lower and much safer doses. I want to thank Pao again for the very nice introduction. Pao joined my lab many years ago as a postdoc and looked at the list of genes which are involved in the RASopathies and took a special interest in RIT1, member of the RAS superfamily, which is a bit odd in the sense it doesn't have a cat's box and has some other very interesting features. And through a really brilliant piece of molecular biology and biochemistry, Pao showed the mechanism by which mutations lead to accumulation of RIT1 and cause subtypes of Noonan syndrome. So these mutations that caught his eye do not occur in the classical codons 12 and the 61 you see in, say, KRAS, but they occur at different parts of the protein. And again, Pao was able to figure out that these mutations make RIT1 resistant to degradation by another protein, LZTR1, which is itself a Noonan syndrome gene. And this is the model that he came up with, in which LZTR1 normally ubiquitinates and degrades RIT1, but mutations in RIT1 make them resistant. Loss of LZTR1 obviously has the same effect. And this little pedigree beautifully shows the power of that model. So in this pedigree, both mum and dad had mutations in LZTR1, it's recessive, no real phenotype, but each of their 4 children, by bad luck, inherited both mutant alleles. So they have no LZTR1 or very little LZTR1, and RIP1 accumulates and causes Noonan syndrome in these individuals. So Power's gone on to figure out more about the mechanism of RIP1, and it turns out to activate the MAP kinase pathway indirectly. RIP1 binds to RAF kinase through the RBD, as I mentioned previously, like the other RASopathy proteins, but doesn't actually activate it. So it basically passes on the RAF protein that's been recruited to RAS itself, which can then finish off the activation process. So it kind of facilitates RAF activation, a very novel mechanism, which I think is really important in understanding the whole process of RAF activation. Now, LZTR1, also very interesting, was already known to be involved in disease. Mutations in LZTR1 are thought to be involved in schwannomatosis. That's a bit odd to me because this is not a disease characterized by activation of the MAP kinase pathway, I don't think. So I'm not really sure exactly how it works here. Power may well know better than I. LZTR1 is being seen as a resistance mechanism in lung cancer, and mutations in splicing enzymes affect LZTR1, which causes accumulation of RIP1 and contributes to leukemia. Also, for those of you who like looking at DepMap, as I do, you'll notice if you look at genes whose knockdown correlates with knockdown of RAF1, the genes that correlate most are genes involved in RAS MAP kinase pathway activation, SOS and SHP2 and KRAS and so on. The genes which anti-correlate, which have exactly the opposite effect of knocking down RAF1, the negative regulators of RAS pathway we know about, Sybil, SPRED2, RASA2, GAP, and NF1, but the strongest correlate, anti-correlate, is actually LZTR1. So knocking down LZTR1 has absolutely the opposite effect as knocking down RAF1 out of all the genes that were interrogated in these DepMap, some 80,000 genes. So this tells me that LZTR1 has another very powerful effect in suppressing the RAS pathway, probably beyond its effects on RIP1. And it just says there's a lot more to be learned about this really interesting gene and pathway. Another set of genes involved in Noonan syndrome, RAS, RAS and RAS2, also very interesting because these proteins, as I mentioned earlier, are known to activate PI3 kinase, not the MAP kinase pathway. So how can they be involved in Noonan syndrome? This is not really their business. So these are 2 papers which identified RAS2 in Noonan syndrome, and I believe representatives of both these groups are here online for this Zoom meeting. So hi. We came across RAS2 from a completely different project when we were looking at the regulation of SOS proteins. Now, when Curian and colleagues solved the structure of the catalytic part of SOS, which regulates RAS GTP exchange, he found, as expected, a RAS protein in the exchange site, NT, in the process of going through exchange, but also saw another RAS protein from his in the test tube, which he defined at another site, which he defined then as a allosteric site. So in this model, RAS GTP has an allosteric effect which activates SOS to enable it to activate itself. That's the model which is in literature. That model didn't really make any sense to me, so we decided to test it in a sort of unbiased way by looking for any other proteins that might bind at this allosteric site that would make kind of more sense from the signaling point of view. And my student Armando Martinez did a mass spec analysis of proteins that bind to the wild type SOS, but not to the mutant catalytic allosteric site, and found RAS2 as the major hit. And he followed this up with a pull down of various members of the RAS family in their active states as per in Noonan syndrome, and found indeed that RAS2 binds strongly to the wild type SOS, but not to the allosteric mutant. So this makes this a candidate for the bona fide allosteric regulator of SOS, which makes more sense to me than RAS itself. But then Armando showed that if you dump this mutant, the activating mutant of RAS2 onto cells, it does indeed activate the MAP kinase pathway very strongly. But it does absolutely nothing in cells in which we've knocked out both alleles of SOS1 and SOS2. So these double knockout cells made by Matt Sale in my lab completely unresponsive. So RAS2 works through SOS to activate the MAP kinase pathway. And we've shown here, well, it's a work from Andy Stevens at the Frederick, that RAS2 is indeed a really potent allosteric regulator of SOS1 in the test tube. So we believe now that RAS proteins have a role in regulating this pathway. The problem is now we don't understand how RAS proteins are regulated normally. They're not clients of SOS. They have other gaps and gaps, presumably, which respond to signals, which may be important in gating the whole of this signaling pathway, but this is all a work in progress. So another complex, which again, Pam mentioned in his introduction, is the MRAS SHOC2 PP1C complex. This has become of more interest recently, but it's pretty cool biochemistry. So this is a basic pathway of how RAS proteins activate RAF. So RAF exists in the cytoplasm in an inactive state, held inactive through these molecular handcuffs called 1433 proteins that bind to these phosphoserines. Now, when RAS is active in the GTP state, RAS binds to the RAS binding domain, which is exposed here to the inactive RAF and recruited to the plasma membrane. And now the cysteine-rich domain plays a very important role in moving away from the kinase and into the plasma membrane where it interacts with specific lipids. And this interaction allows opening of the RAF protein to allow it to dimerize and activate RAF kinase. Simplified version, but this is basically the sort of bottom line as to how RAS activates RAF and the important role of the cysteine-rich domain that I mentioned earlier. However, this phosphoserine here on RAF1 can still rebind rebind SHOC2 and turn the protein off again. So this is presumably a reversible reaction. So to prevent that happening and to make sure the reaction goes in the right direction, another complex comes along to dephosphorylate 259 and push the protein towards the activation state. This is the complex that Pablo Rodriguez-Viciano discovered when he was in my lab many years ago as an effector of MRAS, SHOC2, and PP1C, and a graduate student in his lab at the time, Lucy Young, now in my lab, in collaboration with my lab, showed that in this pathway, mutations occur in MRAS and in PP1C and in SHOC2 in Noonan syndrome. And each of these mutations serves to increase the potency of this complex to make it more efficient at activating the pathway. Also, Bruce Gelb and others have shown that that mutations occur in RAF1 itself around this residue, and now we know in the cysteine-rich domain, which help open up the RAF kinase to make it more likely to go into the direction of RAF activation. So a lot of interest in getting the molecular details of this. We still don't have a complete description, but we are getting there. We have a structure, we and three other groups, including Jason Kwon, maybe at the meeting today, solved the structure of MRAS bound to SHOC2 and PP1C by cryo-EM and by conventional crystallography. And this gives us ways in which we can think about targeting this complex. And indeed, the group at Novartis have developed compounds which affect the interface between SHOC2 and RAS and can potentially be used as therapeutics in cells in which this pathway is particularly important. That is, subsets of tumor cells and maybe also certain RASopathies. But life is never that simple. Although we discovered SHOC2 and PP1C as an MRAS associated complex, we now know that other RAS proteins such as KRAS and NRAS cannot also interact with SHOC2, particularly the Q61 mutants, as Lucy Young has shown here. So in tumor cells with Q61 mutants of RAS, these proteins more likely bind to SHOC2 and PP1C than MRAS itself, which is probably not highly expressed in these tissues. So now we're trying to figure out which of these is the most important and what is the structural basis of this allele-specific interaction between these different KRAS alleles and SHOC2. So surprisingly, wild-type KRAS does bind to SHOC2. Q61 mutants also bind, but G12 mutants do not. And we do have a structural basis for this, which we can present maybe at a later point in the future. Okay, so last sort of vignette I want to discuss is the role of different gaps in regulating the pathway. These are the gaps which we think play a role in regulating canonical RAS proteins. NF1 is the major one, as I'll show you, with its partner in crime, SPRED1 and SPRED2, which form tight complexes and work together hand in hand. RASA2 and RASA3 also have a role in regulating wild-type RAS proteins, as does SYNGAP1, but specifically in brain, as you all probably know. So these are the structures of the RAS family, again, and their involvement in human disease. The original RAS gap, we first purified P120 RAS gap, is not involved in cancer, but it is involved in capillary malformations, arterial malformations. RASA2 is often deleted in melanoma, where it's quite an early, potentially primary mutation in certain types of melanoma. And again, SYNGAP is heterozygous in some very severe types of autism. NF1 you all know about for its role in NF1 and in sporadic cancers. RASA2 has been identified as a Noonan syndrome gene. I'm not sure how widely this has been reproduced in bigger cohorts, but these mutations have been reported in the gap domain of RASA2 gap as well. Now, the consequence of knocking out NF1 in cells in culture is pretty dramatic. We've looked at the consequence of deleting NF1 in various cell types. These are melanocytes. I'm looking at the response to FGF and IGF-1 in this example, but we've done many others. So in green, this is the response to the wild-type cells to growth factors. You get a baseline map phospho work, nice bump when you add, in this case, FGF, and it's back down to baseline again. But in the absence of NF1, you have a higher baseline, a higher peak, and a higher overall activation of the pathway, which takes a long time to come back down again. And we see this with FGF and IGF-1 and other growth factors as well. So the total amount of phospho work induced by a given amount of growth factor is massively increased when you knock out NF1. We knock out RASA1 and RASA2. You also see a bump in RAS GTP binding and phospho work compared with wild-type cells, in this case in response to neuroregulin. But losing NF1 is much more dramatic. And the combination of all three gives you a somewhat increase as well. So here again, normalized to normal cells. This is the effect of knocking out RASA1 and RASA2 in these cells. Knocking out NF1 gives you a much bigger response, and you add on top of that RASA1 and 2, and you prolong this response a little further. So again, loss of NF1 in multiple cell types in response to multiple growth factors has a tremendous impact on overall signaling output. Now, to understand how things work, we've pursuing the sort of biochemistry of neurofibromin for quite a long time. One step forward we made was to show that the SPRED1 protein, which is the mutated in Legius syndrome, forms a complex with neurofibromin through the EVH1 domain of SPRED1. This has a KIT binding domain and a sprouty domain, which are involved in membrane localization, we think. This protein is essential for neurofibromin to work on RAS. Actually, my student worked on this program after hearing Eric Legius's talk at the RASopathy meeting in Berkeley many years ago that Kate Rowan organized. And we went back and thought, wow, that was cool. Let's see if the mutant forms of SPRED1 give us some insights on how things work. And she found by mass spec that neurofibromin binds to wild type SPRED1, but not to pathogenic mutants. So SPRED1 does indeed recruit neurofibromin to the plasma membrane. A more recent experiment here from Lucy Young shows that if you transfect into cells SPRED1, you increase the level of neurofibromin at the plasma membrane. And if you overexpress also KIT receptor, you further increase the activity as shown here. So SPRED1 does have a KIT binding domain, and we think all these proteins form a complex in the plasma membrane to really fully attack RAS and turn it off efficiently. And we're trying to get structural analysis performed on the complexes between SPRED1, NF1, and KIT and other receptors. So this kind of summarizes what we think about the recruitment of neurofibromin to interact with RAS in the plasma membrane. We and 3 other groups solved the structure of neurofibromin. Turns out to be a itself, as you all know, a gigantic protein of over 2,800 amino acids. We showed in collaboration with the group at Frederick National Lab run by Dom Esposito that this is a dimer protein. It's in its native state. It's an obligate dimer, as if it wasn't big enough. And this dimer is basically head to tail configuration, and pretty much the whole protein is made up of bundles of alpha helices, as shown here. So it's been transformed from a long stick of unknown function to a very tightly wound dimeric protein. And we think that this structure allows signal input to affect the ability of the neurofibromin protein to interact with RAS and turn RAS off. This in red here is the gap domain, which was here in orange, now here in red. Obviously, two of these in the dimer. Most of the structures that were solved of neurofibromin show that the gap domain is somewhat occluded, so that RAS can't actually interact efficiently with the gap domain. And we already knew actually from biochemical analysis that full-length recombinant neurofibromin is pretty poor at interacting with RAS by binding or by gap activity. So what we believe is the case is that in response to certain signals, maybe phosphorylation events or maybe other small molecules, that the neurofibromin protein opens up to allow RAS to get access to the gap domain. So that's one aspect of RAS of NF1 regulation, which we're working on right now. SPRED1 is important to get the whole complex into the plasma membrane, as shown here. NF1 also has a Sec14 domain, which when it gets to which can also interact with the membrane. And this whole complex can interact with the KIT receptor and other receptors probably to sort of really underscore that this is an extremely complicated process involving multiple proteins to orchestrate precise regulation of RAS to turn it off under the right conditions in response to the right signals. And we still have a lot to learn about this and are attacking this from the biochemical and the structural biology point of view. So if we think about ways we can the neurofibromin is regulated, well, first of all, the spread proteins are direct targets of the MAP kinase pathway. So high MAP kinase signaling leads to production of spreads, which couple up with neurofibromin to turn RAS off. So there is a very simple feedback pathway, which is definitely important in certain cell conditions. Both neurofibromin and spread proteins are substrates of multiple phosphorylation events. One of these we discovered a while ago prevents SPRED1 binding to NF1 and allows RAS to accumulate in the GTP state in tumor cells with mutant EGFR receptor, but there are other ways of opening up this protein. So we are now performing a high throughput screen for small molecules which can increase neurofibromin binding to RAS proteins in my lab at UCSF, and a parallel lab is working on a similar assay at NCATS at the NCI. So this, we hope, will help understand how NF1 protein is regulated in normal cells, and maybe controlled perhaps in heterozygous states in pathogenic situations. So, thinking about therapeutic approaches, several groups, including Lucy Young in my group, have actually suggested that the major client of neurofibromin, of the canonical RAS proteins, is actually KRAS more than HRAS or NRAS. So these publications make that point. And this is an experiment from Lucy in which she's done pull downs of a whole family of RAS proteins and measured binding to neurofibromin. And you can see that KRAS proteins bind much more tightly than HRAS or NRAS. Don't really understand why, because the interface between these seems to be extremely similar, but there's obviously a significant difference. Also, if you knock out NF1, it's KRAS that comes to life more than HRAS and MRAS, as shown by Yadina Samuels and others. So this led Lucy, when she first came to my lab, to propose that pan-KRAS drugs could be useful in treating NF1, where KRAS would be superactivated. Now, at that time, there weren't any pan-KRAS drugs, so it's a nice idea, but we couldn't really pursue it. But now, as you all know, there are multiple pan-KRAS compounds already in the clinic, including one from BridgeBio, we call 818, which is a pan-KRAS compound, which is now advancing through clinical trials. So this idea has been actually reduced to a beautiful proof of concept by Nancy Ratner, who has And I don't believe she talked about it at this meeting, but in previous meetings she has shown data in which the Boehringer Ingelheim pan-KRAS compound has shown to be as effective as MEK inhibitors in mouse models of NF1, and we believe that pan-KRAS compounds will not have serious side effects, unlike MEK inhibitors, because HRAS and NRAS will be spared, and therefore we'll see less systemic toxicity. TBD, but that's our hope for the future. And with that, I'll stop, and I'll thank my colleagues in my lab and my great friend and colleague from the past, Pau, and the grad student he worked with, and Antonio. Matt Sale in my lab has done a lot of the knockout work, and Lucy's a lot of the work on RASopathies. Amando did the source work. This is my lab hard at work back in San Francisco. And I want to credit Kate Rowan, who got me interested in the whole RASopathy field many years ago when we collaborated on looking at the mechanism of RAS pathway activation based on her brilliant insights in Costello syndrome and CFC. So with that, I will stop and be happy to take any questions.
Thank you, Frank. That was great as usual. We have a couple of questions. Can you say your name when you ask the question so Frank can know?
Know how to respond.
Hey, Frank, Tamar Green from Stanford. Thank you.
Hey, how are you?
Much for coming. Good. I have two quick questions, and sorry for being more basic about it. One question is, in the case of RASopathies, do we activate the other pathway or mostly activate the RAS MAPK pathway? And my second question is, can you speak a little bit about, you mentioned that SYNGAP is mostly expressed in the brain. I'm curious what you think about other RASopathies and NF1 in terms of specificity to the brain.
Yeah, great questions. Yeah. With respect to the first question, as far as we know, the activated alleles of the RAS that we see in RASopathies pretty much only activate MAP kinase. One possible exception might be the Q62L mutation of RAS2, because of the small number of alleles that are being studied, that one is particularly severe. So it may be that in addition to MAP kinase, it also activates PI3 kinase, as I would expect. We haven't actually looked at that specifically in the context of RASopathies, but I wouldn't be surprised if some of these proteins that overlap into other swim lanes are not, probably not as cleanly overlapping as one might think. So I would leave that open, and others may have tested that possibility. But I would say that that's a sort of outlier. I think mostly they just activate MAP kinase. Yeah, the question of NF1 and SYNGAP is sort of beyond my pay grade, but it's in your wheelhouse. So I've always assumed that the issues of behavioral problems and learning disabilities you see with some cohorts of NF1 people.
Please. Frank,
there's a fire.
Okay.
Let's wait for a couple of minutes because sometimes I'll just turn it off.
Okay, shall I keep talking?
Has been reported in the building. Please leave the building by the nearest exit. Do not use the elevators.
Well, it looks like there's an evacuation.
Okay. Well, this happens a lot when I give a talk like this. So. Well, thank you, everybody.
Right. Let's thank Frank at least. So thanks, Frank.
Okay. Enjoy the rest of the meeting.
Thank you.
Great to see you, pal.